In a spinal cord injury, the initial trauma is just the beginning of a devastating condition. Damaged axons are blocked by glial scars and inflammatory cysts and start to degenerate in the hostile environment. Some neurons with severed axons may, however, survive. If these injured axons could be made to regrow to their innervating targets, a certain level of functional recovery might be possible. During development, and whenever neurons must grow later in life, this growth occurs at a highly motile axon tip known as the growth cone. According to recent Axon Guidance studies, the cone's pathfinding is guided by a number of mechanisms, including both attraction to and repulsion from the extracellular matrix, and long-range chemoattraction, mediated by various neurotrophic factors secreted from target cells. In an attempt to simulate these axon guidance mechanisms, we are developing a tissue-engineered scaffold system. A concentration gradient of nerve growth factor, or NGF, is immobilized in a bioactive 3D gel. The gradient slope can be controlled. Lab-grown neural cells are placed on the matrix. After the cells have grown into the matrix, we use directional data analysis theory to investigate the pattern of outgrowth. Within the gel, a coordinate system is defined with its origin at the center of the cell body. Normalized point set data for the positions of the neurite tips can be obtained from the coordinate system. These lines represent the location of neurite tips and show their angles relative to the direction of the gradient. Numerous samples from each gradient are statistically analyzed to test for directional bias, to see which NGF gradient slopes will most effectively elicit axonal guidance. Our experimental results show that we can achieve directed neurite outgrowth by varying the slope of the NGF gradient. We believe this system can be combined with other strategies to develop a regenerative therapy to guide axons across the gap, potentially allowing severed axons to regenerate and reinnervate their targets.